Hello students, today we are going to discuss about one of the practical, that is the culture media. So these culture medias are required for the growth of microorganism from infected material to identify the causative agent. So the basic constituents of this culture media includes like water, electrolytes, peptone, meat extract, blood or serum, and then agar. So in case of water, it will provide source of hydrogen and oxygen, electrolyte like sodium chloride and other electrolytes. Then peptone, it is a complex mixture of partially digested proteins. It is obtained from lean meat or other protein materials such as heart muscle, casein or fibrin, usually by digestion with proteolytic enzymes. Then we have the meat extract, which contains protein degradation products, inorganic salts, carbohydrates and growth factors. Then we also have blood or serum. These are used for enriching culture media. Usually 5 to 10% defibrinated sheep blood is used, and in some cases this serum is used. Then the agar, it is prepared from seaweed. It is available either in long shreds or powdered form. So this agar does not provide any nutrition to the bacteria but acts as a solidifying agent only. So coming to the types of the media, we have different types of media based on different requirements. So it is based, medias are classified based on physical state like liquid, semi-solid, then we have solid media, and then based on the presence or absence of molecular oxygen and reducing substances in the media like aerobic and anaerobic media. Then based on the nutritional factors, we have simple media, complex media, synthetic media. So we have this one special media like enrichment, selective, differential, indicator, transport and sugar media. So these all comes under special media. Okay, so the next we have is the first media we are going to discuss is the simple media. Out here they are the medias which are routinely used in the lab for diagnostic purposes. Okay, so example like nutrient agar, nutrient broth, these are the simple media. So they contains peptone water and meat extract around 1%, and when we add glucose to this nutrient broth, it becomes glucose broth, and when we add 2 to 3% agar to this nutrient broth, it becomes nutrient agar. So these are the simplest and routinely employed medium in the lab for diagnostic purposes. Okay, then the next we have is the complex and synthetic media. So these complex and synthetic medias, we will need extra care, we will need to add extra things in this case. So the complex media, they are they have added ingredients for bringing out certain properties or providing special nutrients required for the growth of the bacterium. And in case of synthetic media, these are prepared from pure chemicals and the exact composition of the medium is known. So these are used for special studies such as metabolic requirements, and the example under this we have is the Dubose medium. So the first media we are going to discuss under complex media is the enriched media. Out here you will see we have example like blood agar, chocolate agar and Loeffler serum slope. So this enriched media, when basal medium is added with some nutrients such as blood, serum or egg, it is called as enriched medium because they are enriched with extra nutrients. So example I have mentioned, we have blood agar, chocolate agar and then Loeffler serum slope. So the first picture we have out here is the blood agar. Here blood is added to nutrient agar It may be used for growing a number of bacteria, but one specific example we have is the Streptococcus, which requires blood for its growth. Then the second picture we can see we have is the chocolate agar. It is a heated blood agar used for isolation of Neisseria and Haemophilus influenzae. Then the next media, that is the third picture, is the Loeffler's serum slope. Here serum is added for enriching the medium. So this medium is mainly used for the growth of Corynebacterium diphtheriae. So the next we have is the enrichment media. Out here we will have tetrathionate broth. A few examples under this we have tetrathionate broth, selenite F broth, and alkaline peptone water. So this enriched media, out here some substances are incorporated in the liquid medium which have a stimulating effect on the bacteria to be grown or inhibits its competitors. Then this will result in an absolute increase in the number of wanted bacteria related to other bacteria. Such media are called enrichment media. So these are very useful for culture of feces where the non-pathogenic or commensal bacteria tend to overgrow the pathogenic ones. Example we have like Salmonella being overgrown by Escherichia coli. So the first picture or the first test tube we have is the selenite F broth. This has similar action as that of tetrathionate broth. Then the second test tube is the tetrathionate broth. Here tetrathionate is added, which inhibits coliforms which allows typhoid paratyphoid bacilli to grow. Then we have the last test tube is the alkaline peptone water. Mostly this is used for the growth of growing Vibrio cholerae. So that is for the enrichment media. Next media we have is the selective media. Out here we have example like DCA or deoxycholate citrate agar, then BSA bile salt agar, then MSA mannitol salt agar. So these are few examples or few medias under selective media. So this selective media, they contains substances that inhibit all but a few types of bacteria and facilitate the isolation of a particular species. So this media are used to isolate a particular bacteria from specimens where mixed bacterial flora is expected. So in case of DCA, deoxycholate citrate agar, addition of deoxycholate will be there, which acts as a selective agent for enteric bacilli like Salmonella and Shigella. Then in case of BSA, bile salt agar, out here bile salt is a selective agent. It favors the growth of only Vibrio cholera, whereas inhibits the growth of other intestinal organisms. Then the last picture is the MSA, mannitol salt agar. Out here this media is mostly used for it is a selective media for Staphylococcus aureus. To differentiate it from other species, we will use mannitol salt agar. So mostly it is for the Staphylococcus aureus. Okay. Then we have the next is the differential media. This will help us in distinguishing or differentiating the organisms or bacteria. So out here, when a medium contains substances which helps to distinguish different characteristics of bacteria, it is known as differential medium. So example we have like McConkey agar or McConkey's medium. This contains peptone, lactose, agar, sodium taurocholate, and neutral red. So out here we have the sugar lactose. So we will see whether the organism is lactose fermenter or non-lactose fermenter. So if it is lactose fermenter, you will see they will form or produce pink color colonies, whereas if they are non-lactose fermenters or NLF, they will produce colorless or pale colonies. That is in the case of McConkey's medium. Then next we have is the indicator media, which will indicate the color change. Okay, so this media contain an indicator which changes color when a bacterium grows in them. So that is why it is known as an indicator media. So example we have, if you see the first picture, it is Wilson and Blair medium. Then we have McConkey's agar again. This also comes under indicator media. Then we have the blood agar. So in Wilson and Blair medium, out here Salmonella typhi grow as black colonies on Wilson and Blair medium containing sulfide. Then in case of McConkey, due to fermentation of lactose, we have discussed just now, due to fermentation of lactose, there is acidic pH which forms the pink colonies in the presence of neutral red indicator. Out here neutral red is the indicator. Then we have blood agar. It is an enriched medium, but bacteria lysing red blood cells give clearing, or we also called it hemolysis, around their colonies. Thus it is also an indicator medium. So that's all for the indicator media. Then next we have is the transport media. Out here we need this transport media in some cases when we grow the delicate organism like gonococci, which may not survive the time taken for transit or may be overgrown by non-pathogenic bacteria if we left them outside for a longer period, if the culture is not proceed as soon as possible. So there might be overgrowth of non-pathogenic bacteria. So for that purpose we will need transport media, and if we want to transport it from one hospital to the other or from one clinic to the other, for the case where we don't have some required materials or reagents for performing the detecting the bacteria. So out here we have example like Stewart's transport medium and buffered glycerol saline transport medium. So these are few examples of transport media. So the first picture is the Stewart's transport medium. Out here this medium, it is a non-nutrient soft agar gel containing a reducing agent to prevent oxidation and charcoal to neutralize bacterial inhibitors. Then it may be used for growth for organisms such as gonococci. Then in case of buffered glycerol saline transport medium, these are mostly used for enteric bacilli. So that is all about transport media. Next we have is the sugar media. So this sugar media, they are mostly needed or required for they are helpful in the identification of bacteria. So in microbiology, the term sugar will denote as fermentable substances, which means we can check whether the bacteria is it can ferment the sugars or not. They are like sugar fermenter or non-sugar fermenters, right? So few examples like sugars like glucose, lactose, sucrose and mannitol, which are routinely employed for fermentation tests. These are few sugars which we use routinely in the lab. So we will also need a Durham's tube. It is a small tube which is kept inverted in the large sugar tube to detect the presence or absence of gas or gas production. Then the colorless medium will turn pink with production of acid by bacteria, and gas production is indicated by gas bubbles accumulated in this Durham's tube. So few examples I mentioned here is the His serum sugars and the triple sugar iron, or we also call it TSI. So these are few examples of sugar media. So that's all about culture media. I hope it is clear. Thank you.